Hi, everyone. Really excited to be here today to talk all things fiber photometry. And just a quick introduction before we get started. I did my PhD at UNC Chapel Hill, looking at the role of neuromodulation in anxiety and fear before transitioning over to the industry side of things. And I'm currently a scientist here at MBF Bioscience, and I'm the product manager for our fiber photometry product line. So I've seen all there is to see when it comes to photometry. And I spent many years in the lab doing hundreds and hundreds of photometry experiments. And now that I'm on this side of things, I get to work with labs every day. And I've seen all the possible things that can go right with photometry and all the possible things that can go wrong with photometry. So today I'm going to be telling you all the things that I think that you should know getting started, and all the little tips and tricks that you can't really get from reviews and other things. So I hope this will be a helpful resource for everyone. And before we start, I know there will be questions that come up, so feel free to ask via the Q&A button at the bottom of your screen. Feel free to ask as they come up, and then I will take a look at the end and answer as many as I can. So today I'm going to start out by doing a quick introduction to fiber photometry. I'm going to talk about what types of signals you can record with FP, and then I'm going to transition over to talk about considerations for experimental design and best practices. And then finally, common mistakes that I see people make a lot and how to avoid them. So to start off, what is fiber photometry? Photometry is an in vivo imaging technique that allows you to visualize neural activity in real time by measuring fluorescence as a proxy for that neural activity. So with fiber photometry, what you need first is a fluorescence sensor. And there's tons of different types of fluorescence sensors that are used for FP, but the commonality between all of them is that they have some resting fluorescence properties, and upon binding to their ligand of interest, their fluorescence capacity increases multiple times over, and you can record those changes in fluorescence as a proxy for the concentration of your molecule of interest. So with these sensors, they're typically all genetically encoded. What you do is you inject them into the brain, into your region of interest, and if you want to target specific cell populations, you can use Cree-dependent sensor, and then you implant optical fibers in that same region, which gives you optical access to that part of the brain, and then you can record those signals as the animal is behaving. So what you get is something that looks like this. Squiggly traces, lots of spikes. And with photometry, it's quite easy to interpret. Generally, an increase in fluorescence means an increase in the concentration of your molecule of interest. And there's lots of different ways that you can use photometry to try to understand neural activity. So the most common is by recording in cell bodies, but you also can record in terminals if you're interested in looking at projections to other regions. And with the explosion of new neurotransmitter sensors that are being developed, you can now directly monitor neurotransmitters and signaling molecules. So there's a few key advantages of fiber photometry over other neuroimaging techniques, namely being able to record in freely moving animals. So unlike something like two-photon imaging, you don't have to head fix your animals, which makes it really easy to combine with behavior and other data streams. Almost always photometry is done with some other data stream, usually behavior, but you can also combine with, say, in vivo ephys, pharmacology, optogenetics, and things like that. With FP, you can get cell type specificity, and you can also get concentrations of specific neurotransmitters using these specialized sensors. And one thing that's really cool about fiber photometry is that you can record simultaneously in different wavelengths, which allows you to record multiple signals at the same time. So this example here is from a study that was recording both dopamine and acetylcholine activity at the same time using different wavelengths. So it's quite a powerful tool, and gives you a little bit more specificity than you would get with, say, ephys, where you can't tell what types of cells you are recording from. With photometry, because it's kind of low footprint on the skull, You can record from multiple regions at the same time. You're really limited more by how many things you can put on the skull than how many things you can put into the system. So you can record bilaterally, you can record upstream, downstream. It's quite a powerful way to understand how signals propagate in the brain. With FP, you can also get millisecond temporal resolution. And this is particularly important when we talk about neurotransmitters, because until now, you were kind of limited by, you're on like the minutes to 10 minutes time scale with techniques like microdialysis. So this now gives you much better temporal access to neurotransmitter dynamics. But that does not come at the expense of the length of the recording. So with FP, you use such small amounts of light that you actually can record for really long times without having any impact to your sensor and your brain tissue. So we've had labs that can record for weeks at a time, looking at circadian rhythms. So FP gives you kind of the best of both worlds, being able to do some really tightly time-locked things to look at discrete signals, and you can also look at how those signals change over time. And so it's really easy to do things like this, or you can correlate behavior and activity. And in this case, what you're looking at on the top is fluorescent signal, and on the bottom, locomotor activity. And so it gives you this really powerful way to try to understand exactly what your cells are doing. And the nice thing about FP is that you can see something like this in real time. It's not like you have to process all of your data to be able to understand what's happening. You can see this in real time as you're sitting there recording, which is quite satisfying. Okay, how does an FP3002 system actually work? So with our system, excitation light of various wavelengths comes from LEDs within the system. That light gets pushed through an objective and then through a patch cord and implanted fiber into the brain, where it's going to excite your sensor of interest. The emission from that sensor is then going to travel back up the patch cord, back into the objective. And once it's in the system, it's going to get separated spectrally from the excitation light. So you're just looking at the signal coming from your sensor. And then that signal is further split by wavelength. So if you're recording with both green and red sensors at the same time, those signals get separated out so that you can look at them independently. They go through a series of lenses and filters before getting projected onto either side of a CMOS camera sensor, which is going to quantify that light that's coming through. That signal then goes to a computer for further processing and integration with things like timestamps. Every photometry system is a little bit different, but these are kind of all the things that they have in common. The difference generally is the timing of the light delivery and the type of detector, but it's all kind of the same basic components. So what types of signals can you measure with fiber photometry? So the classic indicator that's used for photometry is GCamp, which is a genetically encoded calcium indicator. You might see it abbreviated as a GECI. GCamp works by essentially undergoing a conformational shift upon binding to calcium. So there's two main components of GCamp. The first is a binding domain that actually binds to calcium itself, and then this circularly permuted GFP. So what that means is the GFP molecule is kind of split this way, and when calcium binds, the entire sensor undergoes a conformational shift. That GFP comes together and now has a much higher capacity for fluorescence. And so you can record those changes in fluorescence and interpret them as changes in calcium levels. So GCamp typically is expressed in the cytosol, and so what you're measuring is the intracellular calcium concentration. Why do we care about calcium? With photometry, you can't quite record as fast as voltage changes happen in cells. And so we can kind of get around this by looking at calcium, and we use this calcium influx that results as a result of depolarizations in the cell or action potentials, and we can measure those calcium transients as a proxy for neural activity. And so this is a trace. This was a recording done in Slice, where they were recording both GCamp signal and electrical activity at the same time. And what you can see is that every time the cells spike, you get this blip in your GCamp signal. And you can see all the way down to individual spikes. And then if you get a volley of spikes that happen in quick succession, you get these kind of aggregated peaks that look like this trace on the right here, where you can kind of see the signal goes up and then it decays altogether. So with GCamp, because it fills the cells, you can record both from cell bodies and terminals. And there are also red-shifted calcium indicators like RCamp, JR-Gecko, that allow you to record calcium with a red fluorescent protein instead of green, which gives you the ability to record two different signals at the same time. So in addition to GCamp, there's now been this explosion of neurotransmitter sensors. And this is something that's new since like 2018, and I'm sure you've all heard about it by now. They're very, very important. So currently there's sensors available for all kinds of neurotransmitters and signaling molecules. And now there's tons of variants for each one. And these work similarly to GCamp in that you have both a binding domain and a CpGFP. In this case, the binding domain tends to vary across sensors. So the most common way that these sensors are made are by taking a GPCR and modifying it to insert that CpGFP somewhere into it. And so what happens is as your neurotransmitter of interest, in this case dopamine, binds to the modified GPCR, you get this increase in fluorescence. Not every sensor is GPCR based. Some of them are based on periplasmic binding proteins, but the vast majority are GPCR based. Typically, neurotransmitter sensors are expressed on the membrane. And so instead of recording intracellular levels, what you're recording is extracellular. So what you can see is like the release of that neurotransmitter or signaling molecule. And again, with these, there's a whole variety of colors. There are some sensors that are red, which gives you the ability to, again, multiplex and record multiple signals at the same time. So with photometry, the signal that you're actually recording is different than what you would get with, say, like 1-photon miniscope imaging or 2-photon. But to explain that, I'm actually going to show you some 2P data because I think it really helps to make sense of what it is you're recording with FP. So this is a video of a GCamp recording in Cortex. And what you can see is that the cells have some like resting fluorescence. And then as the cells fire, you get these flashes of green. And so if you were to record or extract the signal from individual cells, what you get is something like this, where you have all of these individual traces where you're isolating the signal from those cells. With photometry, what you're recording looks more like this, where you're just kind of getting a collection of all of the cells within a certain area. In this case, it's generally the area below your implanted fiber. And what you get is this kind of average aggregated signal. And so I've extracted the signals here. And so what you can see is that this like fiber photometry equivalent from this image is that you get the combination of all these different spikes that are happening in different cells, and you get one signal. So you can't get quite as granular as you could with 2-photon. You can't look at like, synchrony, asynchrony, those types of things, but instead you get this kind of general aggregated activity. But the benefit is that you can then record from multiple places at the same time and see how signals are propagating through the brain. So a really common question that I get is how many cells are you actually recording from with photometry? So there's been some really helpful studies done lately that have tried to assess this. So what you're looking at on the left is an image of light transmission within brain tissue. So this is accounting for scattering that takes place when you put light into brain tissue. And so what you're looking at is the collection efficiency as a function of distance from the fiber. So the fiber tip in this case would be right at the top of the screen. And what you can see is that the strongest signal is collected right at the bottom of the fiber. and it diminishes pretty quickly with distance. And in this paper, they did all kinds of different comparisons with different fiber diameter sizes, different numerical apertures. And their conclusion was that your fiber core diameter is the most important determinant of the volume of tissue that you're collecting signal from. The numerical aperture makes somewhat of a difference, but it's subtle. So what we can learn from this is that you want to implant at the same coordinates that you're recording from. One common mistake I see is that people inject their virus and then they implant their fiber like 200 microns above, which I know people do often with optogenetics. But with photometry, you don't want to do that because what happens is you're going to miss your signal. The brightest signal is going to come right at the end of your fiber. So if you're not right in those cells, you're not going to pick up a whole lot. So you always want to implant at the exact coordinates of your target. The other thing that this tells us is that you don't have to worry too much about your sensor expression bleeding into other regions of interest. Even if your GCamp overruns into other structures, it's going to be far enough away from your fiber that you're not going to pick it up. So don't worry too much about making sure that your expression stays exactly just within your structure of interest. That said, putting in a bigger fiber isn't necessarily better. And this is kind of a nuanced idea. So consider first a dense population of cells. If you're recording with a 200 micron fiber, that's kind of the standard starter size that we see people use, you're picking up plenty of cells as is. If you were to use instead a 400 micron fiber, you are picking up more cells, but what's going to happen is your signal is going to increase proportionally with your background. And so what that means is the net effect is zero. Your signal to noise ratio is going to look the same if you're using a 200 micron fiber or a 400 micron fiber. The exception comes when you're recording in a sparse population, or if you're recording with if you're recording from terminals or something like that that are much, much smaller than cells. When you're in a sparse population, your fiber might not necessarily happen to end up on top of a cell. With this, it's kind of the luck of the draw. Even if your targeting is perfect, you're exactly at your target coordinates, if you happen to not end up right on a cell, you might not get much of a signal. So in the case of a sparse population, using a larger fiber can give you a higher chance of landing right on a cell. So my advice to people is if you have a sense of the density of your population, start with a 200 micron fiber anyway. If you know that it's super sparse or if you're picking up a signal that's teeny, try a larger fiber. But keep in mind that you are punching a hole in the tissue. This is an image of a 200 micron fiber, and what you can see is that the damage is non-trivial, and if you're recording in deep brain, you're punching through everything else that's above that. And so if you can use a smaller fiber, it's just going to cause less damage to the brain. So I always tell people start with a 200 micron fiber. If you're having trouble picking up a signal and the histology looks fine, there's no reason to explain why you might not get a good signal, then try increasing your fiber size. But in most cases, a 200 micron fiber is sufficient. And this applies both for rats and mice. I've seen people assume, oh, if I'm doing a rat, everything just needs to be bigger. It's not necessarily true. So always start with the 200, and if you need to, go up in size. You also can go smaller. I've seen people record with 50 micron fibers, 100 micron fibers. Up to you. I don't think it makes a huge difference, but I would avoid going bigger. Okay, considerations for experimental design and best practices. These are all of the random things that I wish I knew in grad school, and I wish I knew going into these things, so hopefully this will provide some helpful background. So the first question I get generally is okay, there's so many GCamps out there, how do I pick one? What I usually tell people is if you're starting new with a fresh experiment, go with the latest. Start with GCamp 8. GCamp is really good. Anything that's like GCamp 6 or later is excellent right now. Like we're at a place where we're just like optimizing, uh, and it's not totally necessary. But what that means is that you can't really go wrong. No matter what GCamp you use, you're gonna get a good signal. But if you're starting new, might as well start with the latest. Within each like GCamp family, there are different variants. Generally, they're called S, F, and M. S stands for sensitive, F is for fast, M is for medium. The trade-off between these variants is speed versus sensitivity. So GCamp 8F, this trace is up here. It has the fastest kinetics, but it's the least sensitive. And so GCamp 8F can be really helpful if you're wanting to like resolve individual events. Say you're doing some kind of operant behavior, the mouse is lever pressing really fast, and you want to be able to see those individual responses. That's a case in which you might want to pick a fast variant. If you're starting with a population that you know is sparse, or if you don't know if you're going to get a good signal, or you're recording from terminals, start with S. So S is going to give you the best chance of picking up a signal. And if you look at the responses to individual spikes, right here in our top trace here with GCamp 8F, you can see like if you have a single spike, you're just gonna get a little tiny blip. You don't really get more noticeable spikes until you get these kind of compound events. But if you look down at the bottom at GCamp 8S, what you can see is it's much easier to pick up those single spike events. So I always tell people, if you don't know what you're looking for, just start with M. Just don't make a decision, start in the middle. But if you want to try to maximize your signal, if you're not sure if it's going to work or not, using an S variant can kind of give you the best chance of picking up something. I wouldn't recommend starting with 8 ever. I don't think it's super necessary. But if you are wanting to look at really fast events and you have a strong, robust population, totally fine to start with 8F. Another thing to consider is your approach for getting that GCamp under your fiber. So if you're using a transgenic mouse, the expression is going to be lower than if you were to use viral expression. So there are transgenic mice that you can get from Jackson Labs that have Cree-dependent GCamp already, and all you do is cross those mice with Cree line, and then you have labeling in your population of interest. This works fine. It's not perfect, but the amount of expression that you get is going to vary a lot by the Cree line that you're crossing to. I've seen expression be better in cortex than in deep brain. So if you're doing like tons of experiments where it's going to save you time to not have to do the virus injection, using an AI-148 can be helpful. But in my experience, it's not going to give you as robust of expression as if you were to do a virus injection. So if you're recording from a sparse population, definitely go with a virus. It will just give you a much brighter, more robust signal and make it easier to record what you're trying to record. For best results, I strongly encourage you to pilot different sensors and compare. A lot of times it's like, okay, theoretically, I would pick this sensor if I'm doing this or this sensor if I'm doing this. But a lot of times there's so many unknowns that you can't really tell what's going to be the best without just testing it. So I always encourage people, it's worth your time to spend 3 weeks, just do a pilot, test different sensors. You can put one on one hemisphere, one on the other, and compare directly. But that's kind of the best way to see what works for you is just to test them. When it comes to neurotransmitter sensor variants, there's a lot more to think about here. The most important thing, in my opinion, is thinking about which receptor the sensor is based on. So with, say, for example, dopamine sensors, some of them are based on D1, some of them are based on D2. This is especially important if you're doing any kind of pharmacology, because often the receptors are sensitive to the same drugs as the endogenous receptors. So say you're wanting to give a drug and see how that impacts dopamine release in the striatum. If you're giving a drug, it might be acting directly on your sensor. So that's something that's really important to think about. The sensor, or sorry, the receptor that the sensors are based on are also going to determine things like affinity, sensitivity, and all that. So that's something to keep in mind as well. This is also something that's more of a nuanced thing, but the expression pattern of the sensor often does mimic the endogenous or the trafficking pattern of the endogenous receptor. So what that means is that the pattern of expression that you get in your sensor is going to look different depending on what the sensor is based on. So I have here at the bottom 3 different images of different serotonin sensors. All of them are sensing serotonin, but what you can see is that the expression pattern varies dramatically. It's not really clear exactly what that means in terms of the signal that you pick up, but say, for example, you're interested in understanding the contribution of serotonin receptor 2C in some behavior. You might want to pick a sensor that's based on 5-HT2C so that the concentrations that it's responding to are similar to the concentrations that the endogenous receptor are responding to, and it will also mimic that expression pattern. This is like kind of a nuanced thing. It's not necessarily something you have to think about, but if you're wondering kind of what differentiates these sensors, this is one of them. Affinity and dynamic range, like I mentioned before, is a factor. The other thing that can be an issue is crosstalk between neurotransmitters. This is most relevant if you're recording dopamine or norepinephrine. They are quite similar structurally, and so the dopamine sensors do have some affinity for norepinephrine. Norepinephrine sensors have some affinity for dopamine. So just keep that in mind as well. The sensitivities vary by variant, so just keep that in mind as you're picking a sensor. But just as with GCAMP, the easiest way to decide is just to consult the literature, see what other people have done in your region of interest, behavior of interest, and just pilot. Theoretically, you can think about the affinity and dynamic range, but often we don't know what the concentration of neurotransmitter is in that region of interest. So the easiest way is just compare them side by side, see what works best for the behavior that you're doing, and then go with that. Red sensors. Okay. My general advice for using red-shifted sensors is that if you're only recording from one population, use a green sensor instead. In general, the red-shifted indicators are much less optimized than the green sensors. The green sensors at this point have been through so many iterations that they're just much more optimized and advanced than the red sensors. The red sensors also, there's also some like issues with stability, and I've been told anecdotally that there's issues with getting those red fluorescent proteins to fold properly at physiological temperatures. So the net effect is that the red sensors are just not quite as good as green. So if you're just recording from one signal, use a green sensor instead. But if you're wanting to multiplex, the options are there. So if you are recording from 2 populations, if you have a sense of which population is going to give you the better signal, use the red sensor for that one. Just give yourself the best chance of success. If you're doing something where you're recording from a neurotransmitter sensor and GCAMP, do your calcium in red and your neurotransmitter in green, because the calcium sensors in general are more advanced than the neurotransmitter sensors. You will also want to pilot the virus cocktail that you're injecting. It can be difficult to get both a green and a red sensor to co-express, and sometimes it takes some dialing in of your dilutions, promoters, things like that. So just keep that in mind. I think it's always worth your time to just do a pilot and see if you get expression. It's much easier than putting the fiber in there and trying to record, wasting time trying to get a signal, and then you do the histology and find out one of your sensors didn't express. So always test. And then another thing that's kind of specific to red sensors is this phenomenon called photoactivation. So with some red sensors, and not all, if you hit them with 488 light, which is typically the wavelength that you would use to excite green sensors, you can get what's called photoactivation, which means you get these signals, you get these spikes that look a lot like actual activity, but they're just driven by the light itself. In this figure that I'm showing you, they're testing at 70 milliwatts, which is like blasting them with light. That's way more light than you would ever need even for opto. But a lot of these sensors show this property at much lower light levels. So keep in mind, if you're recording with green and red sensors together, that you want to pick a red sensor that doesn't show activation at 488, because otherwise you're going to get all these false signals that are not real. So keep that in mind as you go. Okay, let's talk for a minute about controls. This is one of the most common things that I work with labs on, and it can be quite nuanced. So let's start with GCamp. Probably heard of the isospastic point before, but let me just talk about what that is. So with GCamp, it does have some resting fluorescence. It's not like totally dark and then when calcium binds, it lights up. It does have some resting fluorescence levels. So if we look at the excitation curves for GCamp, that is both bound to calcium, this darker trace, and unbound to calcium, this blue trace, what you can see is at like 470, 480 nanometers, where you're typically exciting your green sensor, the excitation that you get when GCamp is bound to calcium is much higher than down here when it's not bound to calcium. And that delta is what allows you to actually record when there's changes in calcium levels. If you look closer to like 420, 425 nanometers, what you can see is that these two curves cross. We call this the isospastic point. So what this means is that if you excite GCamp at this wavelength, the emission that you get is going to be the same regardless of whether GCamp is bound to calcium or not. And so what this means is that you can record at both of these wavelengths, and you get this kind of like convenient built-in control where if there's any fluctuations in your signal, you can attribute them to things other than actual calcium events. So I have some example traces here. So on the top, I have my 470 signal. This is your calcium dependent signal. And on the bottom, 415 nanometer signal, which is isospastic. And what you can see is that there's all kinds of dynamics happening in the 470 that are not happening in the 415, but we have this giant artifact spike that's showing up in both. Because we're seeing it in both channels, we can attribute it to an artifact. So the things that you're going to pick up in both signals are motion artifacts, autofluorescence in your tissue, artifacts from your patch cord bending, all kinds of different things. Theoretically, and again, this is not perfect, but theoretically, in your 470, you're going to also pick up neural activity. Theoretically, you won't pick up neural activity in your 415. And so what you can do is use this control signal to identify putative motion artifacts and then correct them out. This is a property that is somewhat specific to GCamp, though it does apply to some other sensors. So if you're thinking about other sensors, there's a lot more planning that goes into determining proper controls. So some sensors do have an isospastic point. Some of them are published. A lot of them, either those curves don't ever cross, there is no isospastic point, or it's not published. So what I always tell people is consult the literature. And what you're going to look for is a figure that looks like this, where you can see the excitation, the emission curves, and generally they'll point out the wavelength at which those two curves cross. So if your sensor does have an isospastic point, it's a great control. The advantage is that you can record that signal through the exact same fiber as your signal of interest. But the con is that the isospastic point of many sensors isn't 415 or 405, which is typically the wavelength of light that photometry systems are equipped with. A lot of them are closer to 440. Most systems don't have a 440 excitation LED, so you can't always use the isospastic point for control. So there are some other options. My favorite is the mutant sensors. So many neurotransmitters are created with, like, in pair with these what we call mutant sensors. So essentially, these sensors are based on the same GPCR, but there is some mutation made to it such that it doesn't bind to the molecule of interest. And so what that means is you can get these kind of companion sensors that have the same expression pattern, the same optical properties, the same resting brightness, but they're insensitive to your ligand of interest. And so the advantage of this is that you'll get a control signal that's like pretty consistent with the signal that you're getting in terms of brightness. But the con is that you can't then record from that mutant sensor within the same ROI as your actual signal. You'd have to do something like put the real sensor on one hemisphere, put the mutant sensor on the other hemisphere, or just do a control group altogether. If your sensor doesn't have a mutant sensor companion, what you can do is just use a GFP instead. With this, the advantage is that you don't have to have a specific companion sensor for each of your neurotransmitter sensors. And this is an example on the right of what this looks like. This is a norepinephrine sensor compared to a YFP control. And what you can see is that you can use it as a way to just gauge baseline activity. You can record motion artifacts that way, but you won't, it'll be separate from your actual neurotransmitter signal. The con of this approach is that often just like a static GFP is like orders of magnitude brighter than your sensor of interest. And what that means is that it's so bright that any artifact that you get is going to be so tiny compared to it. And so what you can get is a control that looks really flat and a signal that looks all like it's all over the place. And so with this approach, it's really important to dilute your virus to try to match the basal fluorescence levels so that if you do have an artifact from your patch cord bending or something like that, it's going to show up similarly in those two signals. And similar to the mutant sensor, you can't record within the same region. But if you do want that within region control, you can use a red fluorescent protein, or in this case, just a fluorescent protein of opposite or different color than your sensor. So what you can do is just co-express your neurotransmitter sensor in green with a TD tomato or an M cherry, and you could basically do the same thing. This gives you a within ROI control. The only catch here is that you do pick up a lot of autofluorescence in your green channel that you're not going to pick up in your red. You can't avoid this. There's tons of autofluorescence within the tissue. And so what happens is that the shape of that decay is going to be a little bit different between the two wavelengths. So that's just something to consider if you're doing something like, recording baseline activity and giving a drug where you expect a baseline shift in your signal. Using a red fluorescent protein in this case can make it a little bit tricky to interpret those results. There's also another trick that I kind of mentioned before with the GPCR-based sensors. You can give a receptor antagonist for that receptor and theoretically block the activity of the sensor. So this is an example of how this was done. On top, we have our neurotransmitter sensor. When they excite, this is in combination with opto, they can drive evoked release of norepinephrine. And then if they give an alpha-2 antagonist, they can then block that signal. So this is really nice if you're trying to show this population spikes, we give this drug, it goes away. We know that those spikes were driven by binding to neurotransmitter. Okay. With this, you don't have to do a separate animal, but you can't necessarily control for motion as well in this way. And the other con is that you can get off-target effects of the drug that you're giving. So just keep that in mind. Okay, patch cord management. This is something that comes up a lot. With photometry, you have to have your mice tethered, which can be a little bit problematic, but there are ways to mitigate issues with this. But first, I want to talk a little bit about how light propagates through a patch cord. So a patch cord is made up of multiple layers of silica, and those layers have different refractive indexes. So when light travels through a patch cord, it essentially bounces back and forth off of the internal surface of the cladding of the fiber, and it travels up the fiber. The reflection within the patch cord depends on the angle of light at which the light hits this cladding. So if the angle that it hits the cladding at is less than the critical angle, that means that instead of reflecting back into the patch cord, it's going to exit. So what this means practically is that if your patch cord bends, it's going to change the angle of the light, which means that you will lose some signal. So generally, if you were to just like record a signal and bend your patch cord, you'll see those fluctuations within your signal. So it's really important to minimize the bending and looping of your patch cord in order to record a stable signal. And this is done by strategically hanging your patch cord. This is something that's super important to setting up a fiber photometry experiment. So the best way to do this is to secure it in some way above your behavior chamber. Don't let it just flop over the side. Stick it up to put it on a ring stand, tape it to the ceiling, put it on the wall. Doesn't matter, but you want it to be hung above the animal. Sometimes this means drilling a hole. If you're using like a Met Associates sound attenuating cabinet where there's no hole, you might have to drill one in the top. You also want to leave proper slack in your patch cord. So you want the animal to be able to travel to all corners of your box, but you don't want it to be drooping so much that the mouse is going to be able to chew on it. That is the fastest way to damage your patch cord, is to let a mouse pull it down, bite it, it's done. It's made of glass. It's very easy to break. Another thing that can help a lot is hanging your patch cord in a way that allows it to rotate. So that as the animal moves, your patch cord will twist. That is why with photometry, you generally use a longer patch cord than you would for opto. And the reason for this is that if you have a long patch cord, you can absorb those twists over the full length of the patch cord. And so what that means is you want to hang your patch cord in a way that allows it to twist. My favorite way to do this is by using a paper clip. So instead of just taping the patch cord directly, because that's not going to let it twist, just take a paper clip, bend it open, tape the paper clip up, and then feed your patch cord through it. And that means as your animal rotates, it's going to be able to spin and absorb those twists across the full length of the patch cord. You also want to make sure to remove anything that the patch cord can catch on. So if you're recording in the home cage, take out the shelters, take out the food hoppers. You don't want your patch cord to get stuck on things. Additionally, if you're using a patch cord that has more branches than regions that you're recording from, coil them out of the way. You don't want them to be flopping in front of your animal because, again, it's going to get in the way. It's going to get chewed. Just coil those up, cover them up, keep them out of the way, easy. A really common question I get is about rotary joints. You can use a rotary joint that will essentially allow your patch cord to twist infinitely without it impacting, without the whole thing twisting. I don't recommend rotary joints with fiber photometry unless it's necessary. The reason for this is every patch cord junction that you introduce into your light path is going to lead to light loss, and you also can get significant light loss through the rotary joint itself. So if you're recording from a really robust, strong population, it might be okay. You might have enough signal left over after all the attenuation to have something usable. If you're recording from terminals or a neurotransmitter sensor, that loss can mean the difference between having a usable signal and an unusable signal. So I always strongly encourage people, start without a rotary joint. And if you find that you're just getting tangled and it's chaos, try it and see, but just keep in mind your signal will look different. Okay, best practices for starting a new experiment. So if you're just getting started with a new experiment, new population, first thing you want to do is pilot your sensors, consult the literature to see what controls other people have used, what acquisition parameters they have used. It's worth your while to test these things before you do your real experiments. The second thing that comes up a lot is habituating animals. The tethering is going to impact behavior. And if you're doing something that's like very sensitive, like operant self-administration, it can be difficult to get the animals to behave while they're tethered. So what you want to do is just habituate them as much as you can. So my favorite way to do this is just take your mice, line them up on a countertop, put them in cages with an open top, and take an old patch cord. You can use a real patch cord if you only have one, but just make sure they don't chew it. But the best thing you can do is just take an old patch cord, cut it off, and just tape it up to the wall and let your animals hang out, get used to being tethered. It will make a big difference for your behavior. You also want to get them used to being scruffed, and this is a hill I will die on. You do not need to put your animals under to tether them. Habituate them to being scruffed. It's not super difficult to attach a patch cord. You scruff them, you wiggle it down on there, that's it. If you're having to put them under every time you're connecting and disconnecting, you're going to have to wait for them to recover before you can do your behavior. So invest time in habituating your animals. It will make your life easier. And save your broken patch cords. Never throw them away. The next thing that's important that people often don't think about is figure out how you're going to synchronize your photometry and your behavior or whatever else you're doing. If you're tracking within EthoVision or AnyMaze, there are ways to get those systems to talk to each other. So make sure that you're thinking about those things as you're setting up. That way you can get everything very nicely time locked. Okay. If you're starting a brand new experiment where you haven't dealt with this population before, I strongly encourage you to invest time in getting to know your population. The best way to do this is just put the animal in an open field, put them in a home cage, and just record video and see what happens. The most important thing that you can do is look at your signal, look at points of interest, go back to your video and see what the mouse was doing at that time. See if you can identify patterns, and then do the opposite. Look at your video, point out an interesting behavior, and then go look at your signal and see what happens at that point. This is going to give you the best sense of what your population is actually doing. So invest time to just see what's happening before you do your very specific experiment. Another thing that I tell people is just try presenting different stimuli and see what happens. The easiest thing to do is just like an air puff. Get one of those little compressed air cans, just give them a little puff of air, see how the cells respond. You can also do things like reward, give them a piece of cereal, give them a piece of sugar, whatever, see what happens. Throw them in a box with another animal, give a drug, do a foot shock. Just see what you can get your signal to do. I always tell people it's easiest if you have a positive control, but try to see if you can make your signal go up, see if you can make it go down, see if you can make it go quiet. These three traces at the bottom here, these are all different responses from the same population of neurons. And what you can see is that not every response is going to be a spike. That's typically what people think of when they think of calcium indicators is, okay, I'm looking for spikes. Not necessarily. You might get something that's more like a baseline shift, and you might get a dip in your signal. So try to find things that make your signal go up and things that make your signal go down. It's going to make your life much easier when you're trying to figure out what's happening and what you're looking at. Okay, final section of this talk is common mistakes and how to avoid them. These are things that I've seen people do many times. It can really impact your recording, so these are the things to avoid. Okay, the first is light transmission issues. This is typically the most common thing that's causing your signal to be noisy or flat. Often this is due to a broken patch cord. So patch cords are made of glass. They are bendy, but they're fragile. The most common type of damage I see is from the patch cord getting super looped, which causes it to crack internally, which means that your light is going to scatter. That's going to cause significant artifacts in your signal. So good patch cord management is super key. One thing that you can do to kind of gauge the health of your patch cord is when you get a new one, put a known amount of light into it, record with a power meter how much light is coming out of it, and keep those records. If all of a sudden one day you have to crank your power up twice as much to get the same output, that's a really good indicator that there is damage to your patch cord. So it's worth your while to just note that, and that way, if you're seeing funky things, you can test your patch cord and it could be the cause. Another thing that's really common but super easy to fix is if there's debris on the top of your fiber or on the end of your patch cord, any dust or bedding or anything that gets in there is going to either cause scattering of light or absorption of light. What I recommend is as you plug in each animal, just give the top of those fibers a quick wipe with either like Kim wipe with water or an alcohol swab or something just to get the dust off of it. It can make a really big difference. Another really common mistake is cleaving fibers poorly. So with fiber photometry, you want your implanted fibers to have a really clean, flat cut on them. This comes down to how you cut those fibers. If you cut it poorly, the end is going to be shattered and you're going to get weird scattering of light, and it's going to be hard to get a good signal. So if you are cutting your own fibers, test them before you implant to make sure that you're not getting significant light loss. That's super, super important. Another common thing I see is that people use patch cord and implants that have mismatched optical specs. So you want to make sure you match the core diameter and the numerical aperture. And the last thing is rotary joints. Avoid them if you can. Start without, add it in if necessary. If you're not picking up a good signal without one, don't add it in. It's not going to make anything better. The other thing that's really common is light leaks. So in general, if you see light coming out anywhere in your light path, that means that you have room light that's leaking in and it's going to show up in your signal. The size of a signal you pick up from the brain is like nanowatts. Overhead lights are watts. So even like a tiny bit of light from a computer screen will impact your signal. There's different reasons why you can get light leak. One common thing I see is using white ceramic for rules. People use these white fibers often for opto, which is fine, but with photometry, you care about the light that's coming back into the brain. So you don't want to use white ceramic for rules. If you've already got animals where you did it already, my advice is just get like a piece of heat shrink tubing and just thread it on your patch cord so that it like sits down over that fiber and just prevents any room light from leaking in. Same thing can happen if you're using translucent dental cement. If you're using that like classic pink orthojet cement, light will leak in. You'll get impacts from overhead lights as well as like if you lean over the animals, you'll get shadows. So make sure you're using opaque dental cement. If you have translucent dental cement on there already, put a layer of black over top just to prevent that light leak. The other common thing that's common or that happens often is a gap where your patch cord attaches to your fiber. You want to make sure that patch cord is like fully seated on there so that there's no light coming out like in this picture. Same thing if your patch cord gets chewed or something and you see light coming out of the side of your patch cord, it's going to be a problem. The easiest way to test if you have light leak is just flip the room lights on and off while you're recording. If you see a step function like this, it means you have light leak. If your signal is stable, you're good. Okay, last thing, don't start with a complicated experiment. I talk to so many labs that are so ambitious and they want to do this really complicated thing. If you're setting up photometry for the first time, keep it simple. Just start with a positive control. Generally, this looks like using a non-Cree dependent Gcamp. Use a nice bright Gcamp, anything Gcamp 6 or later, and just put it somewhere that we know has baseline activity. Put it in PFC, put it in striatum, put it in the amygdala. Just give yourself the highest chance for success. What you want to see is a signal with lots of peaks, like this one I have here. It's much harder to interpret what's going wrong if you don't have a signal to work with in the first place. This means that if you're starting and you want a positive control, start with cell bodies. Don't start with terminals. The signal is much smaller, and don't start with a neurotransmitter sensor. The only exception to that is D-light, because D-light is like the most advanced sensor there is. But in general, just keep it easy. Get your bearings with positive control, and that way it's much easier to troubleshoot. All right, and with that, I will take any questions that you have. Just 2 quick mentions before we do. We have open office hours every day or every week on Tuesdays at noon Eastern. If you have questions about photometry or anything else, you can sign up every week and show up, and I will be there, as well as our other product experts. Our other introduction is that we have a new light sheet microscope that we just launched recently. It's a super economical benchtop light sheet scope. We're just getting it out into the field now. We're super excited. So if you're interested in light sheet, please feel free to reach out. And I will answer questions. Okay. Is it common to use high-low pass filters in fiber photometry analysis? I've noticed that filtering practices vary between labs. Yeah, this is a good question. Fiber photometry analysis is very much still the Wild West. There's not a ton of norms out there. What I would say is that some systems sample super fast, like in the kilohertz range, which is like orders of magnitude larger or faster than what you would need for your actual sensors. Um, in those cases, it can help to filter because if you're seeing oscillations in your signal that are happening at like 1 kilohertz, you know it's not due to your sensor because the sensor is not that fast. In that case, low pass filtering can help. Um, in general, I recommend not filtering. Um, with like if you're recording at like say 90 hertz, 60 hertz, something that's like closer to the actual kinetics of your sensor, you kind of by default filter out some of that high frequency noise. But it does vary by sensor. The best thing to do is just see what other people have done with that sensor in the literature and then decide based on that. Okay. Does GCamp have some level of fluorescence activity even when the animal is sedated? I ask because it should be possible to record from the fiber during surgery so that once the fiber hits the right area in the brain, there should be a slight increase in fluorescence signal. Yes, it does. GCamp does have some resting fluorescence. It varies across like variants. But yes, in general, there is always some background level, and you can do exactly that. If you're doing photometry guided surgeries, what you want to do is like very slowly lower down your probe, like go faster until you get to like, you know, couple hundred microns out, and then go slowly. And what you can do is just record and just watch your signal go up and then try to find the peak of that. The signal will look a little different. You generally don't get peaks when the mouse is under, though it does depend on the population, but you should have enough background that you can do your targeting that way. Okay. Okay. I get really high expression levels with my virus. Is bigger better in this case, or does it just increase background? This is a really good question. Um, Photometry is less sensitive to the absolute brightness of your signal than, say, for example, like 1 photon or 2 photon. If you're trying to like resolve individual cells with 1p, 2p, you care about not having a ton of background, but with photometry, it doesn't really matter as much. You tend not to max out your signal. So it doesn't make a huge difference. The place where it can make a difference is with some of the neurotransmitter sensors where the dynamic range is small. So if you have a ton of expression and your baseline is really, really high, it can be difficult to pick up small fluctuations on top of that. And in that case, diluting your virus can help. But in my experience with GCamp, it's like not as much of a deal. So GCamp, fine. Neurotransmitter sensor, you might want to consider diluting. Okay, let's see. Since some peptides, for example, oxytocin and vasopressin, are promiscuous and act through the same receptor in some brain regions, example via oxytocin receptor, if I use a grab sensor for oxytocin in this region, I'll in fact measure both oxytocin and vasopressin with this sensor. Did I get that right? Depends on your sensor. If the validation was done thoroughly, there should be some assessment of the binding affinity to various neurotransmitters. I've seen this happen a lot with norepinephrine and dopamine. The more like optimized sensors are often optimized for picking sensors that have much higher sensitivity to target your target neuromodulator. But there is some crosstalk, but it just varies by variant. So what I would do is just consult the literature and see if they've done those tests and pick one that has the biggest difference between the two. Okay, we're about at time. So if you have to go, feel free to hop off. I will answer a couple more questions while we're here. Let's see. Is there a gold standard for fiber photometry analysis regarding delta F over F or a z-score? This is a really good question. A lot of people show things as delta F over F. It's fine. In my opinion, if you're going to aggregate activity across a cohort, it's best to z-score. You can be as consistent as possible with your injections, your placement, you're always going to have variance in the signal to noise ratio across animals. If you're like quantifying your, say, like your peak height and you're using delta F over F, that's going to vary a lot across mice just based on your fiber placement. And so what happens is if you have a mouse where the fiber ended up like perfectly on top of cells, you have a super bright signal, it's really going to skew your average. So what I say is if you're going to do any kind of cross animal comparisons, Z-score it first to give you some level of standardization and then go from there. I default to Z-scoring all the time. I just think it's easier to use. It's easier to compare, but it's not critical. If you're just looking at like the trace and you want to see how these cells are responding, you can use delta F over F or Z-score. It doesn't make a huge difference. Okay. What is the difference between raw and isospecific data? This is a good question. So the isospecific theoretically is not going to pick up your calcium signal. So in your raw calcium data, you're going to have a combination of both artifact and signal. In your raw isospecific, you theoretically are not going to get that signal. You're just going to get the background fluorescence and artifact. And so what you can do is like use that to deduce if your signal that's showing up in your raw 470 signal is an actual neural event or not. Okay. Oh, this is a good one. Okay, what's your take on recording recent versions of iGlue sniffer and iGABA sniffer at terminals? What would be your advice when using them? With GABA and glutamate specifically, because there's so much GABA and glutamate happening at any given time point, it's easier, or you'll get a cleaner signal if you can restrict the expression with some kind of like Cree driver. So in my experience, if you just like put a iGABA sniffer in a region and try to record, there's so much asynchronous GABA happening that what happens is your signal just averages out to be noise. It helps a lot to improve the signal to noise if you can restrict it in some way. It gets kind of tricky because if you're just looking at a postsynaptic response, like you might not always have a target population that you care about. In that case, even just like diluting it can help. But yeah, specifically with GABA and glutamate, isolating is going to help a lot. And another thing with the glutamate sensors is that's actually one that's an exception when it comes to speed. The glutamate sniffers are super, super fast. So in that case, you want to really up your acquisition frame rate. I would take a look at the literature, see what other people are doing, but typically you need to go like 3 to 4 times faster than you would for GCAM. Okay. I had what I think is a good signal in newly developed sensors, but after euthanizing the animals, I could not detect the virus sensor expression. Any idea what could be the reason? Using a standard PFA transcardial perfusion. Yeah, this is a good question. It can be really difficult to interpret what you're looking at if you don't have strong virus expression. It's often confusing whether you're looking at noise or you're looking at signal. In my experience, if you can see a real signal through your fiber, you'll see the expression in the tissue. And often, like, it's easier to see the expression in the tissue than it is to see it through a fiber. So if you're not seeing it afterwards, even without staining, you should be able to see it. So if you can't see anything, it probably means that you're not picking up a real signal. Yeah, if you can see it in Vivo, you should be able to see it afterwards. If you're not seeing it afterwards, it's probably not there in the first place. You can always stain. Just stain. You don't have to have like a sensor specific stain. Just stain for GFP. It should work. But yeah, in general, if you can't see it, it's probably not expressing. Okay. All right. I am going to go ahead and close down the questions. But if you have any lingering questions, please feel free to email info@mbfbioscience.com. We also have some user forums where you can ask questions, but we're always here as a resource. So if you've got more FP questions, please feel free to send them our way. Thanks so much, everyone.